Yeah, but this one is up. This comes apart.
Oh.
So how about it?
Oh, this one's working now. Maybe.
Wait tonight. I'm reading something. Yeah, not clear now.
It's too small.
Further will be bigger, man.
Yeah.
Better now or wrong?
Maybe. No, that one. I'm sorry.
Yeah. But I think maybe this will cover it.
I would put anything.
Is it? I'm looking more for these.
Do you guys all have a copy of this? Yeah. No? Does anyone else need a copy? Let me take attendance real quick. Can you tell me your names? Starting with, I don't know. Joshua. David? Oh, David Malonzo? Okay. And then Eve. Yes. Sorry? Oh, Alice. Okay. And then Crystal Kira. Kira. Catherine. Okay. Okay, so you guys had the test number 5. So we'll go over this, and then score it together, and then, so that the office can have your scores. Let me pull this up on. I think this is 2018. I think. Yes. Okay. I think. Does Daniel normally make you guys submit your thing online? Or no? No. Okay, let's go for it. Remember questions 1 and 2, it's like 40 to 50% usually of your grade. So just make sure it's like overall going to be a little bit less than a quarter of your overall score, right? So you just want to make sure for number 1 and 2, right? You want to kind of put in the most amount of effort. Make sure your answer is really thorough. So let's go over it. Okay, polar bears are highly adapted for life in cold climates around the North Pole. Brown bears, black bears, and pandas are found in warmer environments. Researchers collected complete mitochondrial DNA sequences from several populations of bears and constructed a phylogenetic tree to represent their evolutionary relatedness, right? So, based on this, right, just from reading the question or the information that they provided you, right, without even looking any further, right? Right? Um, and is inherited. Anything? How? What? Or yeah, this is one of those things that you should just have memorized. Because remember, mitochondrial DNA is inherited a little bit differently from your chromosomal DNA, right? Mitochondrial DNA is always inherited from your mom, right? So it's always maternal inheritance, mother to child, right? So automatically when you see mitochondrial DNA, right, that is what you should think about, right? What you should think of, right? Phylogenetic tree, we know, right? It could be looking at a certain trait. It could be looking at, right? Here we're looking at mitochondrial DNA, so it's showing where all of the divergences happened, right? They give you a little timeline here in hundreds of thousands of years, right? Okay, so let's look at our questions. A researcher studying adaptation in bears sequenced a nuclear gene encoding a lysosomal trafficking protein list in polar bears, brown bears, black bears, and panda bears. There are 7 inferred amino acid substitutions that are found only in polar bears. Mutations that cause similar substitutions in the human list protein are associated with Tradek-Higashi syndrome, an autosomal recessive condition in which pigment is absent from the hair and eyes. Researchers used the inferred amino acid sequences to build the distance matrix shown in Table 1. Okay, so you see your amino acid differences, right? Just the numbers. And then we know that polar bears have, yeah, that amino acid substitutions that causes this in humans, right? Okay, so part A, use the phylogenetic tree in figure 1 to estimate the age in hundreds of thousands of years of the most recent common ancestor of all brown bears. Identify the population of brown bears to which polar bears are most closely related based on the mitochondrial DNA sequence comparison. Identify 2 populations whose positions could be switched without affecting the relationships I illustrated in the cladogram, right? Um, when you see questions like this, identify, um, right? You really just need to identify. Right? They don't ask you to justify, explain. So if it just says identify, you're lucky. All you really have to do is identify. 1 sentence should suffice. If you feel like you need to add something else, you can. But again, based on your kind of the bolded words, right? You should be able to just do that, right? Right enough to satisfy that. Yeah. Okay, so let's estimate the age of the most recent common ancestor of all brown bears. So if we look back at our cladogram, right? Brown bears, brown bears, brown bears, right? So where would be the most recent ancestor? It would be here, right? Right, because we have brown bears here, brown bears here, brown bears here, right? These two, most recent ancestor would be here, and then when you combine it with the European brown bears, it would be here. So I don't know, what is that? This is like 350, right? Thousand, right? So it should be like 30, what? Maybe like 32, 30, or 33, right? Looks about right. Um, they'll probably give you a range, right? But probably 330,000 years ago. They want the age in hundreds of thousands of years, so years ago. I'm assuming, let me pull up this scoring guideline so we can get that. I'm assuming they'll give you a range, right? Because it is not. Yeah, so anything between 30 and or 300,000 years ago, and then 350,000 years ago, right? Should be okay, right? Okay. Identify the population of brown bears to polar bears most closely based on sequence comparison. So that's going to be this first one, right? So polar bears, right, are going to be most similar to your European brown bears, right? They're in the same little branch here, right? So the answer would be European brown bears for this, right? I believe they should both be 1 point for each of the um ones that you got. Let me see. Yeah, so 1 point for each of the bolded words, right? So estimate the age, right? That's 1 point, right? Identify the population, 1 point, and then identify two populations whose positions could be switched without affecting the relationships illustrated in the cladogram, right? So when you look at this, right, these two are in the same clade, so you can switch these around, doesn't matter. You could also switch these around, right? So either European brown bears and polar bears, or Western North American brown bears and Asian brown bears, right? You can pick either these two or these two, and that would be 1 point, right? So part A, total 3 points. So make sure you guys are scoring. Um, right? So 1 point, 1 point, 1 point. Okay, part B. Construct a cladogram on the template to represent a model of the evolutionary relatedness among the bear species based on the differences in list protein sequences, right? Circle the position on the cladogram that represents the outgroup, right? So what is the outgroup? The one that has the least, right, amount of least amount of similarities. So most number of differences, right? Okay, so let's look at that, right? So they give us this, right? Your outgroup is going to be this leftmost uh, branch, right? So where should we have everything? If we look, black and brown bears only have one, right? One amino acid difference, right? So most likely they're going to be the most related, right? So we have black, brown, right? Again, this position, right, can be switched, right? And then let's see, we have panda, and then we have polar that have to be together, right? If you look at your difference between your polar bear and your panda bear, right? There's 40, right? Panda versus black and brown is 33 and 34, right? So if we're looking up and down here, right? Um, we have more similarities with the panda than the polar bear, right? Between your black and brown. So panda would go here, and polar bear would be your outgroup. Right, so when they ask you to circle your position on the cladogram that represents the outgroup, that would be your polar bear. Okay. I'm assuming that would be at least 2 points, right? So polar, panda, black, brown. Again, you could write brown and black. It does not matter. And points. One point, right? So 2 points, right? Okay, a student claims that mitochondrial DNA sequence comparisons provide a more accurate phylogeny of bear species than do list protein sequence comparisons. Provide one piece of reasoning to support the student's claim. Okay, so um, right, if you look at the first piece of information that was given to us, right? So the phylogenetic tree, and then we had an amino acid sequence comparison, right? Um, so it's two ways of representing the same amount of information, right? Trying to figure out um, how related, right, the different species are, right? Where your common ancestors lie, right? If you look at kind of this, right, phylogenetic tree and the cladogram that we just made, right, um, you kind of get two completely different uh, graphs, right? Um, so what could be a piece of reasoning to support that the mitochondrial DNA sequence comparison is better? Anyone have a suggestion? Anyone? No, it's too early. No. Oh, yeah. Yeah. Right. Yeah. So that would be a possible answer, right? Other possible answers could be mitochondrial DNA sequences are more comprehensive, right, versus one protein, right? Um, but yeah, I don't know what else. I don't know, like, if you guys or have questions about like, does this work or something like that, you could throw it in the chat for people who are online. Or if you guys are like, is this answer okay? Then that's fine too. Um, right. So another way of saying that, right? Mitochondrial DNA looks at multiple genes, right? Right, for many proteins, right? Whereas your list protein sequence is just looking at one protein, right? Anytime, right, you do a kind of comparison of one protein, right? Uh, the problem can kind of be, is this protein sequence, right? Or will this protein sequence be representative of your species as a whole, right? Um, yeah, so that's always the question that you have. Um, I don't know. Let's see. So I believe this should be 1 point, possibly two. Yeah, 1 point. The three kind of ones that they give here doesn't necessarily mean it has to be one of those uh, three, right? Genes show more variability in nucleotide sequence than proteins do in your amino acid sequence, right? So essentially, your mitochondrial DNA is going to uh, have more variability, right? Mitochondrial DNA genome contains multiple genes versus one list gene, right? So it's the whole comprehensive, right? Where you have multiple, you're comparing multiple genes, right? Phenotype associated with the list gene is under strong selection, right? So what they're saying, right, regarding that is the list protein, right? Um, uh like basically is under strong selection, meaning it is like causing what, right? It's why the reason why uh your polar bears, right, essentially are going to be white, right? Assuming because for the human list protein, right, it causes your loss of pigment from hair and eyes, right? Now you wouldn't find, right, that specific kind, or you wouldn't want that specific kind of mutation in your black, brown, possibly even panda bears, right? Okay. Okay, part D. A researcher genetically engineers a mouse strain by deleting the mouse list gene and replacing it with the polar bear list gene. Predict the most likely difference in phenotype of the transgenic mouse strain compared to the wild type mouse strain. Justify your prediction. Okay, so predicting, right, what would happen if we replaced our mouse list gene with the polar bear list gene, right? We know what the polar bear list gene causes, uh, or what a similar substitution would cause in humans, right? So you can assume that is what would happen, right, in your mouse strain as well, right? So possibly you would have a loss of pigment in the fur and eyes, right? Justify, right? Again, what I just said, right? You can just basically write from what here, right? Um, you have that similar, uh, you find that similar phenotype in humans with comparable substitutions, right? So that would be your justification. I'm assuming 1 point for each again, right? So for the prediction and the justification, right? So 1 point prediction: mouse fur and or eyes will not have pigment or reduced pigment. Either one works. The fur will be white or lighter, right? So any of that should be okay. 1 point. And then your justification: the polar bear list gene protein is associated with a lack of pigment, white hair, or the mutated human list gene protein is associated with a lack of pigment in hair and eyes. So again, you just need to tie it back to the fact that either the polar bears or humans with that same gene, right, um, resulted, right, in the lack of pigment in the hair and eyes, right? Okay, part E. Describe how the mutation in the list gene became common in the polar bear population. If the list gene were the only determinant of fur color, predict the percent of white offspring produced by a mating between a polar bear and a brown bear. Okay. Um, right, so how the mutation in the list gene became common in the polar bear population, right? This is basically asking for definition of natural selection, right? How did it become common, right? Remember, um, what was I gonna say? Anytime, right, frequency of a particular trait changes, right? We call it evolution, right? Allelic frequency, microevolution, right? Macroevolution, speciation, right? So, yeah, remember natural selection. Anytime, right, you have to talk about natural selection, right? Um, AP Bio loves just using two things to describe natural selection, right? It's your selective advantage. Advantage leads to differential reproductive success, right? So how did this mutation in your list gene become common, right? It provided a selective advantage leading to differential reproductive success, right? Um, so more of the white polar bears survived, right? And so again, right? This is why you have let's see, you have more, right, of these guys, right? So I'm assuming again this E described part is going to be 1 point, right? Okay, so if the list gene were the only determinant of fur color, predict the percent of white offspring produced by mating between a polar bear and a brown bear, right? So this is the only determinant, right? So if you look at your polar bears who have that 7 amino acid um, difference, right? And your brown bears who don't, right? What would be the percent of white offspring, right? You would not be able to produce any white offspring, right? It would be 0%. Yeah. If this was the only determinant of fur color, right? So I'm assuming 1 point, 2 points for each of these, right? 2 points, 2 points, or 1 point, 1 point. Yeah, it's a total 2, right? So record your score out of 10, right? And if we don't have any questions, I'll move on to the second one. Any questions on this? No? Okay. Okay, so we have the next one. Some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways. Cells normally produce a steady supply of inactive caspase 1 protein. In response to intracellular pathogens, the inactive caspase 1 is cleaved and forms an active caspase 1. Active caspase 1 can cleave two other proteins. When caspase 1 cleaves an inactive interleukin, the active portion of the interleukin is released from the cell. An interleukin is a signaling molecule that can activate the immune response. When caspase 1 cleaves gasdermin, the N-terminal proteins of several gasdermin proteins, N-terminal portions of several gasdermin proteins associate in the cell membrane to form large non-specific pores. Researchers created the model in Figure 1 using data from cell fractionation studies. In the experiments, various parts of the cell were separated into fractions by mechanical and chemical methods. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table below shows the presence of known proteins in specific cellular fractions. Okay, so if we just look at this, right, following, right, the information that were given to us, right? So inactive caspase, right, when you have some sort of infection, right, is activated by cleavage, right? And what happens here, right? Basically gets activated, and then what does your caspase 1 then do? It's going to then activate interleukin, right, which is a signal that is going to basically fire up your immune response, right? And the second thing that it's going to do is it's going to activate essentially gasdermin to make a pore, right, in your cell membrane. And then here, right, cell fractionation, right? So you're looking, right, which fraction, right, has what particular protein, right? So you can kind of look, right, based on what you have in your fraction, you can kind of tell, right, what part of the cell you're looking at. Okay, so describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell, right? So if we look here, right, if we inhibit step 3, right, so we're basically blocking step 3, from happening, right? And they wanted to know what was the, my memory is trash, formation of pores and release of interleukin, right? So if we inhibit step 3, right, then our gas dermin wouldn't be cleaved and we would get no pores, right? It would just stop this whole process, right? Does it affect, right, activation of interleukins? No, right? Again, separate process, right? Because in order for, yeah, that's it, right? So your two-part answer, right? It would prevent the formation of pores, right? And then, but your interleukin would still be activated, right? Now, because it is a describe question, right? I would explain, right? I wouldn't just write it would do this and that. I would try to provide somewhat of an explanation, right? Because you still have active caspase 1, right? So your interleukin would still be activated. However, if you inhibited step 3, right, that would prevent your gas dermin from being cleaved to provide, to make, right, N-terminal gas dermin, no pores would be formed, right? Something like that. I believe should be 2 points. We'll see. Yeah, 2 points, right? Pores will not form, release will not be affected, right? Interleukin release will continue. Right? Okay, part B. Make a claim about how cleaving inactive caspase 1 results in activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason to support the student's claim. Okay, so basically they're asking you, why does cleaving, right, or essentially, right, if we look at this step right here, right, how does removal of this portion here, right, how does that activate caspase 1, right? Easy answer, right? Just a very generic answer should be fine, right? Caspase 1 is an enzyme, right? Enzyme means it's a protein, right? What do we know about proteins? Structure equals function, right? Structure of a protein and function of a protein is very closely tied together, right? So by this cleavage here, right, we changed essentially the structure, right, of our protein, which would thus change, right, which could possibly, right, affect the function of our protein slash enzyme, right, which would cause it to go from inactive to active form. Other answers like that little dot, right, could be some sort of um, inhibitory portion, right? Yeah. Okay. Um, okay. So the student's claim, right? Should, oh, sorry, that first one should be 1 point, right? Student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection, right? Provide one reason to support the student's claim, right? So, right, anything along the lines of, right, if you have inactive, right, caspase, caspase 1, right, when you do have some sort of infection, right, all you have to do is cleave that, uh, right, cleave it in order to activate it, right? Versus having to go through the whole transcription translation process in order to make caspase 1, inactive caspase 1 in order to cleave it, right? So again, right, yeah, it just is, uh, it is just much simpler, right? Again, because the alternative would be transcription translation, right? And then turning that polypeptide chain or amino acid chain, right, into a protein, which is going to be much quicker, right? So it makes sense for your cells to kind of pre-produce, right, caspase 1 in order to be ready for a quicker response, right? When you get an infection, right? Okay, so claim, right? It removes an inhibitor, repressor, inhibitory domain of a protein. It changes the shape or protein structure, right? So either of those work. 1 point. Reasoning, cleaving a precursor protein molecule is faster than making one upon infection. Cells don't have to wait for transcription and translation, protein synthesis, right? So 1 point for your claim, 1 point for your reasoning, right? Okay, part C. A student claims that the I don't know what that is. B protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. Identify 2 fractions where N-terminal gasdermin would be found in cells infected with pathogenic bacteria, right? Okay, so they claim that this protein is in the cytoplasm until you need it for transcription, right? So how would you look at that, right? So this protein is an immune response protein, right? You find it, okay, this first one is your control, right? Your whole cell sample, right? Now you find this same protein, right, in fractions 2 and 3, right? So fraction 2, right, what else do you have? You have DNA polymerase, right? What do you use DNA polymerase for? DNA replication, right? Where does DNA replication occur? In your nucleus, right? So we know fraction 2 is our nucleus, right? And then you also see here, right? You have GAPDH, which is a glycolytic protein, right? Glycolytic glycolysis, right? Where does glycolysis occur? From cellular respiration, your cytoplasm, right? So fraction 3, cytoplasm, right? So this would provide evidence, right? That, oops, I don't know why I circled these. You see the immune response protein in both your nucleus and cytoplasm, right? Which could provide evidence for that. So, right, you see it in fraction 3, right? So you see it in your cytoplasm, right? Which would support that student's claim, right? 1 point for that. And then identify 2 fractions where N-terminal gasdermin will be found in cells infected with pathogenic bacteria, right? So again, same thing, right? You find it in fraction 2. Sorry. Two fractions where N-terminal gasdermin would be found, right? So N-terminal gasdermin, right, is going to be found where? In your cytoplasm and on your cell membrane, right? If you look at figure 1, right? So N-terminal gasdermin, again, cytoplasm and cell membrane. So which fractions represent your cytoplasm and your cell membrane, right? We already know, right? Fraction 3 is your cytoplasm, right? What is your other one? Is going to be fraction 4, right? Sodium potassium pump, right? Is found on your cell membrane, right? So again, right? This fraction 4 represents your cell membrane. Yeah. So we have those. Yeah. So fraction 3, fraction 4 is where you would expect to find N-terminal gasdermin, right? Based on figure 1. Okay. So I'm assuming that would be 3 points, right? Justify and then your 2. Yeah. your NFKB and glycolytic enzymes, GAPDH, are found together in the cytoplasm, right? So they're just looking at that fraction 2 right here, right, where you have right here, right? You have GAPDH and your immune response protein together, right, indicating that it is present in your cytoplasm, right? And then your identification of fraction 3 and 4, right, where you would find N-terminal gas stirring, right? Okay, part D. Describe the most likely effect of Gerstmann pore formation on water balance in the cell in a hypotonic environment, right? So hypotonic, right, means what? Right, you have lower, right, concentration of solute, right? Or yeah, and then you have higher concentration here, right? So what would happen, right? Osmosis, right? If you form a bunch of pores, right, you would get what? Hmm? It would dissolve. Ooh, sorry, I can't. Plasmolyze. Okay, plasmolyze is for what is it called? Plant cells, right? Remember plasmolysis that occurs for isotonic, right? Isotonic cells, right? So plasmolysis, right, um, is when your cell wall stays intact, but your cell membrane kind of like moves in towards like this, right? This only occurs for plant cells, right? Plasmolysis, right? Um, so this, actually, I don't even know. Maybe we are. I don't know what type of cell we're even going to be talking about, right? They don't necessarily um, specify which one it is, plant versus animal. Huh? Yeah, yeah. Exactly, right? So you would have right, rush of water into your cell, right? Eventually causing the cell, possibly causing the cell to explode, right? So lysis would likely occur, right? So you would have water moves into cell, right? Okay, so again, part E, right? Explain how Gerstmann pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen, right? So our response, right, just in this image, we had a two-pronged response, right? We form pores, right? What is the result of pores forming, right? Water is going to rush in, likely leading to lysis, right? So if you destroy your cell, right, then your infection is not going to spread, right? Interleukin release, right, is kind of like a warning signal, right? A chemical signal to kind of amplify or how do you start it? Like start, right, your immune response, right? Um, so that you can kind of protect yourself from further infection, right? So Gerstmann pore formation, right, can help you destroy the infected cell, right? Interleukin release is going to help warn, right, neighboring cells um, that there is an infection, right? Let me see. Yeah. So again, explanation, right, for 2 points, right? Again, this should be a lengthier response, right? A more thorough response when they ask you to explain, describe, right? So the points that they gave here: cell lysis destroys infected cells or prevents bacteria from replicating, right? So one addressing the N-terminal Gerstmann, right? Interleukin signaling will stimulate immune cells, components of the immune system, to destroy the infected cells or bacteria, right? So how does interleukin signaling affect the cell, right? Okay. So again, that should be overall 10 points. Okay, number 3. Seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce sticky pollen that is carried by circulating water to female flowers, resulting in fertilization. A researcher claims that mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. To investigate this claim, the researcher set up aquariums to model the possible interactions between the invertebrates and seagrasses. Use the symbols below in the template aquariums to demonstrate the experimental design for testing the researcher's claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Draw the appropriate symbols in the negative control aquarium and the experimental aquarium. Do not use any symbol more than once in the same aquarium, right? Okay. So they want you to draw it in this little aquarium that they have for you, right? So again, how would or what would we have in each, right? So negative control, right? So it would just be your male flower and your female flower, and they only want you to draw one, right? Male flower, female flower, right? And then your experimental aquarium should have all three: male flower, female flower, invertebrates, right? So basically, you would assume, right, if their hypothesis is correct, right, in your negative control aquarium, you should have no pollination, right? And then in your experimental aquarium, you should have pollination, right? Identify the dependent variable in the experiment. Predict the experimental results that would support the researcher's claim that mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. Okay, so what is our dependent variable? What are we looking at? What are the results that we're trying to find, right? You can say pollination, right? Um, you would expect pollination to occur, right? Um, and then what would be the results that would support the claim that these invertebrates can transfer pollen from male to female flowers without water, right? So you without circulating water, right? So in your experimental aquarium, right, with the presence of your invertebrates, you have uh, pollination occur, whereas in your negative control aquarium, you have very little pollination or no pollination, right? So there has to be uh, statistically greater, right, amount of pollination in your experimental aquarium where your invertebrates are present, right? Um, oh, we didn't do points for part A. Probably this is number. So 2 points for your drawing, right? Negative control, 1 point. Experimental, 1 point, right? Okay, so for part B, 3 possible answers, right? So essentially pollination, fertilization, reproduction, right? Either of those are okay, right? If you picked pollination, your prediction should correspond, right? So, like the example that I give you guys was pollination, right? The dependent variable was pollination, right? So you have to have more pollination in your experimental aquarium compared to your negative control. right? If there is no difference, then that means that the invertebrates really did not play much of a role, right? So if you did fertilization, more fertilized plants, flowers, right? Yeah, so whichever one you looked at, right? Make sure you have that. So again, 2 points. 1 point for the identification, and then the corresponding prediction, right? So total, we have 4 points so far. Okay, number 4. The common bedbug is a species of insect that is becoming increasingly resistant to insecticides. Fantastic. Bedbugs possess several genes suspected of contributing to the resistance, including P450, ABC8, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted one or more of these genes in different strains of bedbugs, as indicated in Figure 1, and treated the strains with the insecticide beta-cyfluthrin. Each strain was genetically identical except for the deleted genes and was equally fit in the absence of beta-cyfluthrin. The percent survival of each strain following beta-cyfluthrin treatment is shown in Figure 1. Okay, so if you look at Figure 1, what do we have, right? We have the 5 different strains of bedbugs, right? And we kind of deleted, right? Um, various kind of genes, right? The 3 genes that they suspect leads to resistance, right? So if you look at strain 1, right, again, right, you have uh, all three of the genes for resistance. You see that a very high percentage of those bed bugs did survive uh, that insecticide, right? Um, and then if you look at strain 2, we just deleted P450, right? You see versus strain 1, you see a decrease, right? Statistically significant decrease in your survival of your bed bugs, right? In strain 3, we only removed ABC8, right? Again, you see an even more, right, significant decrease, right? Versus your, again, strain 1, right, which has everything intact, right? Strain 4, right, you remove CPS, right? There is no difference, statistically significant difference between just by removing CPS, right? You can see that here. And then in 5, right, you removed both P450 and ABC8, and you see a very, very significant, right, decline in the percent survival of your bed bugs, right? Um, okay, so identify the control strain in the experiment, right? So we know our control strain is going to be what? Strain 1, right? Strain 1, we did not uh, do anything, right? Um, we did not remove, right, any of these, right? This is kind of our control group, right, to compare, right, what would happen to survivability, right, if we removed one or more, right, of those 3 genes, right? So uh, control strain is 1. Use the means and confidence intervals in Figure 1 to justify the claim that ABC8 is effective at providing resistance to beta-fluthrin, right? Okay, so where did we remove ABC8? That was strain 3, right? So again, how can we justify, right, that ABC8 was successful, right, at providing resistance, right? If you compare 1 and 3, right? Removal of ABC8, we know that this provides resistance because easiest way you can compare the error bars, right? Your your bottom error bar and your top error bar do not overlap, right? Is one way to explain it. Right? And that is to 95% confidence, right? So, yeah, should be 1.1 point. Let's look. Okay, so identification, the control strain, right? Again, that's going to be strain 1. And the justification, right? Your error bars, right, from strain 1 don't overlap with strain 3, right? Mean percent survival of your strain 3 falls outside the 95% confidence interval of strain 1, right? Which is essentially the same thing as saying that the error bars do not overlap, right? Because they tell you that the error bars represent 95% confidence, right? And then strain 3 deletion strain 3 shows a statistically significant difference from strain 1, right? So anytime they ask you, right, if it is a significant difference, right, and they give you a graph like this, it always has to do with your error bars, right? Non-overlapping error bars indicate uh, statistically significant results. Okay. P450 encodes an enzyme that detoxifies insecticides. It's a protein that pumps insecticides out of cells. CPS encodes an external structure protein located in the exoskeleton that greatly reduces the absorption of insecticides. Based on this information and the data in Figure 1, explain how a deletion of both P450 and ABC8 results in lower survival in bed bugs compared with a deletion of CPS only. Okay, so they kind of explain, right? Or they do explain, right? What all of these kind of genes do, right? Um, so they're saying, right? CPS only is this guy, right? Versus 5. So they're saying compare strains 4 and 5, right? Why is this combined, P450 and ABC8 deletions combined, more effective than just a CPS, right? So CPS, right? If we look at strain 4, what does CPS do? It encodes an external structural protein located in the exoskeleton that greatly reduces the absorption of insecticides, right? So it kind of prevents, right, insecticide absorption or reduces insecticide absorption, right? Um, but basically, right, once absorbed, it can't do anything, right? Strain 5, right? Um, P450 again detoxifies, right? So basically, you can just assume that it's probably breaking down, right? This enzyme, right, is breaking down your insecticide, right? And then ABC8, right, makes a pump, right? So it's actively pumping, right? So when you get insecticide exposure, right, P450 is breaking down any insecticide in the cell or in the bed bug, right? ABC8 is pumping it out, right? So therefore, it's going to be much more um, effective, right, than just kind of reducing the amount of insecticide that is being absorbed, right? Because as you can see, right, if we compare strain 1 and 4, right, CPS doesn't seem to be very effective, right? Probably what that means, right, is that even a small amount of insecticide being entering, right, your exoskeleton of the bedbug can be fatal, right? And should be, that was 2 points, right? A was 2 points, so B is probably 2 points as well. Okay, so 1 point per row, 2 points maximum, right? So again, you have to talk about strain 4 and 5, right? So comparison of 4 and 5, right? Um, so again, strain 5, right? Again, you can't detoxify or pump out insecticide, right? All you do is reduce the amount of insecticide being absorbed, right? Again, results in a lower chance of bedbug survival. Strain 4, right? Bedbugs can detoxify and pump out insecticide, results in a higher chance of bedbug survival, right? So just that comparison in terms of the context of what all of your genes do, uh, when you compare strains 4 and 5, right? And that should be 2 points. Right? I think 4 to 6, or sorry, 3 to 6 or 3 to, yeah, something like that is like 4 points, and then it drops to 3 points, I believe. Okay. Number 5. Some birds, including great spotted cuckoos, lay their eggs in the nests of other birds, such as reed warblers. The warbler parents raise the unrelated chicks and provide them with food that would otherwise be given to their biological offspring. Um, a researcher eh? A researcher conducted an investigation to determine the type of relationship between warblers and cuckoos in an environment without predators. The researcher found that nests containing only warblers were more likely to be successful than nests containing warblers and cuckoos. A successful nest is defined as a nest where at least one chick becomes an adult warbler. In some geographic areas, several species of nest predators are present. Researchers have found that cuckoo chicks, while in the nest, produce a smelly substance that deters nest predators. The substance does not remain in the nest if cuckoo chicks are removed. Figure 1 shows a probability that nests containing only warblers or containing both warblers and cuckoos will be successful in an environment with predators. In a follow-up experiment, the researchers added cuckoos to a nest that contained only warblers and removed cuckoos from a nest containing warblers and cuckoos. Okay. I didn't even. Okay. Um, so figure 1 shows probability of nest success in an environment with predators, right? So you can see, right, that the success is greater when you have both warblers and cuckoos, right? Why does that seem likely, right? Um, I mean, uh, you can see, right, like uh, in the second little paragraph there, right? Um, cuckoos, right? Cuckoo chicks, right? Um, help deter predators to that nest, right? Um, so probably that is why, right? I mean, nest success is just defined where you get at least one adult warbler, right? Which isn't that doesn't seem that significant, but that is how they measured it. Um, so yeah, okay. So let's see. Part A. Describe the symbiotic relationship that exists between the cuckoo and warbler in an environment without predators. Okay. So without predators, right? What is happening? Right? Warblers, right? Are they benefiting? No. right? Warblers will only benefit from the presence of cuckoos if there are predators, right? Because the cuckoos, again, like that smelly substance that deters predators, right? But if there is no predator, right, the cuckoo is just what leeching off, right, of the warblers, right? Because the warbler is getting giving, right, the cuckoo babies food that should be given to their offspring, right, biological offspring, right? So symbiotic relationship, um, it would be a parasitic relationship, right? Your cuckoo benefits, right? Warbler is harmed, right? Um, this is not an example. I think in the past, a lot of people put like commensal relationships, right? This is not a case of that, right? For commensal relationship, what needs to happen? One benefits, the other is unaffected, right? This is not a case where your warbler is unaffected because the warbler has to actively raise that um, the cuckoo, right? So the warbler is being affected negatively, right? Um, Obviously, right? Oh, I guess that's part C, right? So we'll talk about that anyways. Okay, so on the template provided, draw bars in the appropriate location to predict the relative probability of success for the nest in the presence of predators where uh, the cuckoos were added to the nest containing only warblers. Cuckoos were removed from the nest containing warblers and cuckoos. Oh, here. Right, so this is the little template that they gave us, right? Um, so in group one, what would happen, right? So we have predators. What would happen when cuckoos are added? When you have only warblers and you add cuckoos, right? You would expect what? Probability would be higher, right? So anything above this should be fine, right? Should be okay. And then for group two, we removed, right? We had warblers and cuckoos together and we removed the cuckoos, right? So it should be something like comparable to here, right? So this one, right? Probability of success goes up. This one, probability of success goes down, right? Um, it should be fine as long as it's higher and lower. Usually when they have these kind of graphs, right? Um, yeah. Um, 1, 2, 3, 4. Right. So I believe part A, right? 1 point. Let's see. Yeah. Cuckoos are parasites, right? They benefit and the warbler is harmed, right? One benefits, one harmed. That's a parasitic relationship. Um, draw bars in the appropriate locations, right? For part B, right? Cuckoos added, right? So in group one, bar must be higher than warbler's only bar, right? So as long as it goes up, should be fine. Bar must be lower than the warblers and cuckoo's bar. So anything lower should be okay. Okay, identify the symbiotic relationship that exists between the cuckoo and warbler in the presence of predators, right? So previously, right, no predators, right? Cuckoo benefits, warbler is harmed, right? Now, when we do have predators, how does that change? Cuckoo still benefits, right? Because they're being cared for by the warbler, right? Warbler also benefits because now, right, the presence of the cuckoos help deter predators, right? So when we have both organisms benefiting, right, that is a mutualistic relationship. So that it would be mutualism, right? So 1 point for part C, they just have it written as what? Uh, Mutualism, both organisms benefit, right? Okay, so again, number 5, also out of 4 points. Okay, next one. Number 6. Cystic fibrosis is a genetic condition associated with deficits in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. In the provided model of a cell, it's like so much drawing in this test for some weird reason. In the provided model of a cell, draw arrows to describe the pathway for production of a normal CFTR protein from gene expression to final cellular location, right? So basically they're saying how do we take, right? How does a gene lead to a protein, right? Okay, so okay, so where does gene expression of our CFTR protein start? In our nucleus, right? And what's going to happen from there, right? Nucleus, right? Again, to ribosome, right? On our endoplasmic reticulum. Remember, we have 2 types of ribosomes, right? We have floating ribosomes and then we have embedded ribosomes, right? Essentially, their role is the same. There is a slight difference. Does anyone know? Difference between your free unattached versus your attached ribosomes? They both do that, right? They both produce proteins, right? Because that's what ribosomes do, right? Same method, right? Translation is still the same, right? Usually it's where your protein is going to end up, right? So not 100%, right? But usually proteins that are made on your rough ER are meant for either your membrane or export, right? And the reason why is it's your rough ER is part of your endomembrane system, right? Your endomembrane system does what, right? It ends with your Golgi apparatus packaging it in a vesicle for, right, export, essentially, right? So typically, if it's meant for either your membrane or export, right, so your membrane proteins or leaving your cell, you will use an attached ribosome, right? Free-floating ribosomes, you could also use it for that, right? It's not like it can't follow that method, but usually for proteins within your cell, right, is what these free-floating ribosomes are going to be used for. Right? Okay, so nucleus to a ribosome on your rough ER, right? Where translation is going to occur, right? Transcription occurs in your nucleus, translation occurs in your endoplasmic reticulum. Where does it go from your endoplasmic reticulum? Your Golgi apparatus, right? What does your Golgi apparatus do? Modify, right? And package your protein, right? And then it's gonna pop off in a little vesicle, right? This is a cis to trans side of your Golgi, right? It's gonna move it into a little vesicle or package it into a little vesicle, and then your CFTR protein can arrive at your plasma membrane, right? Should be 1 point. Right. nucleus, nuclear envelope, endoplasmic reticulum, Golgi apparatus, plasma membrane, right? So there's your pathway here. Yeah, so that is 1 point. Okay, part B. Identify the most likely cellular location of the ribosomes that synthesize CFTR protein, right? So that's what we just kind of talked about, right? So part B, I don't know why they asked B after A, but yeah. Most likely, right? Rough ER, right? Again, right? Um, all you really have to do is identify, right? But reasoning, as I explained earlier, right? Anything that is going to be used for export or on your plasma membrane is very likely going to be produced on your rough ER because it's part of your endoplasmic reticulum, or sorry, endomembrane system, right? Okay, part C. Identify the most likely cellular location of a mutant CFTR protein that has an amino acid substitution into the ATP binding site. Okay, so remember that is essentially right what cystic fibrosis is, right? Where you get a essentially like buildup of like very thick mucus phlegm, right? Um, so where or sorry, most likely cellular location of a mutant CFTR protein with an amino acid substitution in the ATP binding site, right? ATP binding site means what? Very you have ATP binding to allow chloride ions to diffuse across the membrane, right? So what that means is it very likely, right, you're gonna have an ATP binding site at your CFTR, right, channel, right? Um, you're trying to move chloride ions, right, inside. So your binding site, right, is gonna be on your plasma membrane, right? It could be within the cell or outside of the cell. Um, but regardless, right, your cellular location would be at your plasma membrane, right? Remember, any type of channel, right? Channel pump, right? That's all forms or gated, right? This type of gated channel, right? Gated channel, right? These are all forms of active transport, right? Um, the only kind of right type of uh, passive transport that right that wouldn't require any use of ATP, right, would be just simple diffusion, right, or facilitated diffusion in which you use a protein channel like a pore, right. It's kind of always open, right, um, and that would be facilitated diffusion, right. But most transport within or in or out of your cell is going to require some sort of ATP, right, some sort of energy. Um, yeah, but if it's gated, 100%, right? Because you're going to need ATP to open and close that gate, right? Even with your voltage gated channels, right, where they change based on voltage, right? Okay. Like in your uh, brain, right? Your neurons. Um, yeah. Uh, just I don't know like why, but FYI, like they like to ask about cystic fibrosis in your the FRQs, right? Usually they'll give you like all like if it's in a longer question they'll give you a lot of background information so you don't really need to know anything about cystic fibrosis, but it is sometimes helpful to just know like very basic, right? um, because it'll just help your understanding with that question, save you some time, right? But because cystic fibrosis is something that does come up often, right, just keep that in mind. I mean, by the time you guys do all of these um, like practice tests, right? Multiple choice and free response, you will see cystic fibrosis a lot, even in like the multiple choice as well, right? In the tongue solefish, sex is determined by a combination. Oh wait, did I not score this? The last question. Oh yeah. Identification. Sorry for part C. Or, yeah. So 1 point, right? So total 3 points, right? One for the drawing of your arrows, right? Um, one for rough ER, and then one for cell membrane. Okay, number 7. In the tongue solefish, sex is determined by a combination of genetics and environmental temperature. Genetically male fish have 2 Z chromosomes, and genetically female fish have 1 Z chromosome and 1 W chromosome. Right? So male is ZZ, right? Female is ZW. When fish are raised at 22 degrees Celsius, ZZ fish develop into phenotypic males, and ZW fish develop into phenotypic females. However, when fish are raised at 28 degrees Celsius, the Z chromosome is modified as Z star. Z star W individuals develop as phenotypic males that are fertile and can pass on the Z star chromosome to their offspring, even when the offspring are raised at 22 degrees Celsius. A cross between a ZW female and a Z star Z male is shown in the Punnett square below. Okay, let's see. Z chromosome is modified, right? So basically, right, you kind of have different phenotypes here, right? So ZZ is male, right? ZW is male, right? However, you have this weird phenotype here, right, where you have Z star W as male. Oh, wait, sorry, this is female, right? And then based on temperature, this ZZ male can turn into Z star Z male. Okay, so number 7, predict the percent of phenotypic males among the F1 offspring of the cross shown in the Punnett square if the offspring are raised at 22 degrees Celsius. Right, so 22 degrees Celsius, you don't have change in any of your phenotypes, right? So you have this Z star Z is a male, right? This Z star W is also a male, and then ZZ is your male, right? This person is female, right? So 75%. Right? Because remember, ZW individuals are still males when they're raised at 22 degrees Celsius. Okay, should be 1 point, right? 75%. Okay, for part B, at least one Z or one Z star chromosome is necessary for survival of the fish. A researcher crossed 2 fish and observed a 2-to-1 ratio of males to females among the offspring. Based on the information, identify the genotype of the male parent in the cross. Describe one fitness cost to the female of mating with this particular male, right? 2-to-1 ratio, right? When you have a cross, right? That means one of them died, right? Probably this cross right here, right? Because it said you need Z for survival, right? The only way that you would lose or you would have a cross that results in offspring without a Z chromosome would if you had would be if you had this cross right here, right? So if we draw that Punnett square, right? This offspring would die, right? Because again, you need Z or Z star, right? Similar to how humans need an X chromosome for survival, right? Um, not that there would be any way for us to not get an X chromosome, right? And then we had a 2-to-1 ratio of males to females, right? So remember, this is male, and then ZW is female, right? So we have 2 to 1 male to female. Right, so identify the genotype of the male parent. So your male parent has to be Z star W, right? Describe one fitness cost to the female of mating with this particular male, right? The easiest one is what? One of your offspring will die, right? Or 25% of your offspring will likely die, right? Is possibly, right? The, uh, yeah, right? So that's a large fitness cost, right? Um, because without, again, other environmental factors like predators or things like that, right? Um, just genetically, right? 25% will always be predicted to die, right? Yeah. Let's see. Part A, 1 point, right? Identify 1 point, describe 1 point, right? Uh, right, so identify Z star W, right? Description, fewer offspring will develop, survive, 1/4 are predicted to die. Some of her offspring will have the Z star chromosome, all of her male offspring will have a Z star chromosome, right? What is the significance of passing down the Z star chromosome? Why would we care? Why would this be a fitness cost? This third bullet point right here. Why is this a bad thing, right? So if you, right, that means that what? 50% of your males, right? 50% of her son, right? Her male offspring will have this particular phenotype, right? Which will what? When mated with female, will always do what? Lead in 25% of your offspring dead, right? So yeah, that so this is kind of like this is your immediate, right? Fitness cost, right? This is like a future fitness cost, right? Because again, 50% of your sons, right, will always have that ZW, will always be, right, have this particular phenotype, which will result in 50% dead, right? Now, this one, right, this son, right, will create, right, or will pass down this. You could create a son, or you will create a son, right? Probability of having a son like this, right? So that would skip one generation, but you're basically, right, proliferating this particular genotype for your males, right? Um, which again doesn't seem to have much of a disadvantage before, besides the fact that when they reproduce, right, there is a chance that 25% of their offspring will die. Okay. Last one. Acetylcholine. Okay. So acetylcholine receptor proteins are found at the synapse between neurons and skeletal muscle cells. Acetylcholine released from neurons binds to a specific site on the receptor proteins, which causes an ion channel in the receptors to open and allow sodium ions to enter muscle cells. The resulting depolarization of muscle cells initiates muscle contractions. Another molecule, nicotine, can also bind to certain types of acetylcholine receptor proteins and activate the receptors. A researcher is investigating 2 different types of acetylcholine proteins, type 1 and type 2, to determine which stimuli activate the receptors. Researcher exposes muscle cells expressing the different types of receptor proteins to stimuli and observes the results indicated in table 1. Okay, so you have 2 types, acetylcholine receptor type 1 and 2, right? Type 1 responds both to acetylcholine and nicotine, right? Type 2 only responds to acetylcholine, right? So when you expose type 2 to nicotine, right, there is no depolarization, right? Um, why would that be? Or I guess, never mind. So yeah, let's look at A. Describe the difference in the structure and function between acetylcholine receptor type 1 and type 2, right? Tons of different answers that you can kind of give for this particular one, right? Now the difference in function, right, has to do with response to nicotine, right? So the function part is easy, right? So acetylcholine receptor type 2 only can have binding from acetylcholine, right? Nicotine cannot bind, right? Again, structure has to do with the structure of the receptor, right? Type 1 allows for binding of both acetylcholine and nicotine. Type 2 only allows acetylcholine, right? Yeah, I mean, it's literally just what is allowed to bind, right? Um, and then I would just write some sort of description, right? A couple sentences, maybe like 2, 3. So that should be 2 points, I believe. Yeah. Right. So binding sites, different shape, specificity number, differential binding of molecules to type 1 and type 2, right? Receptors activated by one or both molecules, right? Again, function, right? Which is they have the same function, right? Except one is only activated by acetylcholine, right? Type 2 is activated by acetylcholine only, whereas type 1, right, would be activated by 2, right? So if you have nicotine in type 2, right, you wouldn't have depolarization, right? So you wouldn't have a muscle contraction, right? So as long as you explain that, should be fine, right? So binding sites, different shape, specificity number, right? Your function is differential binding of molecules to type 1 and type 2, activated by one acetylcholine molecule or both acetylcholine and nicotine molecules, right? So type 1 for that, type 2 for this one, and type 1 for that. No difference in response, both open channels or both result in depolarization or both cause muscle contraction, right? So whether you have nicotine bind, right, or acetylcholine bind, your results should be the same, right? Like, so for your type 1, whether you activate it through acetylcholine, whether you activate it through nicotine, it should be the same, right? And when type 2, right, is activated, again, same result as when type 1 is activated by acetylcholine or nicotine, right? So that's basically what they're saying, right? Same thing, based on which one you write. So 1 point for your structure, 1 point for your function. Okay, acetylcholinesterase is an enzyme that breaks down acetylcholine in the synapse. Describe the effect of inhibiting acetylcholinesterase on the muscle cells with AChR type 2. Okay. So if we have type 2, right, what would happen? If you basically, right, what is you have, right, your presynaptic cell, right, and then you have your postsynaptic cell, right? In this case, it's a muscle cell. Could be a dendrite of another neuron, right? So at your axon terminal, you have your neurotransmitter, right? In this case, acetylcholine, right? It's released into your synapse, right? You have receptors here, right? Why would you break down acetylcholine in your synapse? What is the purpose of having acetylcholinesterase? think about this muscle, right? So your neuron releases acetylcholine, right? It binds, causes muscle contraction, right? What happens if I leave acetylcholine in that synapse? It's gonna keep contracting. Right, exactly, right? You don't, right? If I leave acetylcholine right here, then it's gonna what? Continuously activate this pathway, right? So it's gonna continuously cause muscle contractions in my postsynaptic cell, right? Usually I don't want to do that, right? If I need continuous contractions, then what should be happening? I would have continued, right, action potentials running through to release neurotransmitters in my presynaptic cell, right? I would be continuously activating this pathway, right? But when I activate it once, I don't want it to stay activated, right? Um, so acetylcholinesterase, right, is an enzyme that basically, right, um, it's going to allow for time to relay the message to my postsynaptic cell, and then any leftover, right, is going to be broken down, right? Um, Not for acetylcholinesterase, but a lot of your, or not for acetylcholine, a lot of your neurotransmitters, they have transporters, right, where you can do reuptake, right? So like one of the common ones, like SSRIs, right? I don't know if you guys heard this, used to treat depression, right? Serotonin selective reuptake inhibitor, right? Because for like serotonin, right? Also, what's going to happen? You're going to release serotonin, right? Some of it is going to be taken back in, right? Any serotonin that is left in your synapse, you're going to take it back in, repackage it, right? So that next time you have serotonin available for release, right? Same thing with dopamine, right? Dopamine that's left over in your synapse, you're going to do reuptake, right? Any leftover dopamine, you're going to package it back in, right? So that next time you have dopamine ready for release, right? So for depression, one common, right, class of drugs is an SSRI, and basically what they do um, is they prevent, right, this drug is going to prevent your cell from reuptake of serotonin, right? Because again, a lot of times depression, right, any kind of neurological problem is caused by problems with signaling. So it's not that your brain isn't releasing serotonin, right, but that serotonin isn't getting across essentially, right? Or the response, right, is dulled down, right, weakened for whatever reason. So you're kind of trying to flood, right, your synapse with serotonin, leave it there longer so that um, to increase the probability or the chance, right, that your serotonin has the desired effect on your mood, right? Also, how drugs work, but different story. Okay, yeah, so that's it, right? Um, so part B, right? 1 point, continued activation, repeated opening of sodium channels, or repeated depolarization or muscle spasms, right? Any of those answers work, right? Um, so what is that? 1 point, 2 point, 3 points, right? Um, so can you guys add up your scores? Um, for if you guys are online, can I think Daniel makes you like post it in the thing, right? Does he make you guys post it too, or he told me to tell what's her name? I don't even know if she's. I didn't think I said Angela. He said to tell Angela. Yeah, so post it. And then we'll kind of let's go over something random, I guess, since we have like 15-ish minutes. So post it right now. I know you guys forget. I don't know where you guys post it. Can you guys post your scores? For number 5. Oh, does he do this? Okay. Test number 5. Please. So add it up and then post it, and then oh, I don't know. What should we go over? Any requests? Huh? Population ecology? Really? Just the whole unit in 10 minutes? Is that what you guys are doing in school? All of you guys? Or are you both them? Do you have a specific question on population ecology? It's easy. Yeah. Let's see. I think population ecology, like ecology, the ecology unit should be relatively easy. Um, Let me see. Like I think uh, there's like sometimes the FRQs, right? The only time it kind of gets weird is sometimes for your FRQs you get calculation problems. Yeah, I mean, yeah, Hardy-Weinberg um, is probably pretty easy, right? Hopefully, yeah. Straightforward. I think like sometimes like people, what is this? Uh, with population ecology, right? They'll ask for like birth rates, death rates, right? Should be pretty easy. Um, I think they also have where's that? Um, Population growth, right? So, um, right, which should be easy, right? It's just kind of all of the formulas are given to you, right? So, sometimes like, right? So, what is the population growth formula? Useful to know, right? It's easy, right? Change in population over change in time is just your birth minus death rates, right? So again, right? Um, if I have, right? If I mean, it depends on what information is given, right? So, but just a basic population growth um, question would be this, right? But then based on whether you have logistic or exponential growth, you have different formulas that you would use, right? What is the difference between logistic and exponential growth? Right, so you have that sigmoidal curve, right? Something like this. Exactly. Carrying capacity. Right, so you have a deceleration, right? You could also have boom bust cycles, right, at your uh, carrying capacity, right, where it kind of shoots above, right, and then it goes down, up and down, right? Okay, and then exponential growth is kind of like if I just looked at it from here, right? Yeah. Okay, so logistic. Ooh. Okay, so logistic growth formula. Hmm? Yes. Right, so dn over dt. I mean, you don't need to memorize the formula. Like I said, it'll be given to you. Oh, sorry, over carrying capacity. right? Um, it will be given to you. You just have to know which one to use and when, right? Um, so this is your R represents per capita growth rate. So this is your maximum per capita growth rate. Right. K represents your carrying capacity. Yeah. And then N is your population size, right? Same. I mean, change in population size, change in time, right? So basically, this is saying like your carrying capacity, right? Basically, how close you are to your carrying capacity is going to affect your growth rate, right? Is essentially what this equation is saying, right? Exponential growth, you don't have that problem, right? So your it's just R max, right? Your growth rate times your population size, right? If your population is increasing at a rate of whatever, right, you just multiply that to your population size, and for exponential growth, that's it, right? Logistic growth, again, based on how close you are to your carrying capacity, right? I don't know, like, what's the problem? Yeah, you guys are gonna kind of do all of these, but in here, okay. So they give you everything here, right? Like all of your equations that you could possibly need, right, will be here, right? Um, you could use it for that, like, you know, at your last, what is that, like five? Are there five or eight? I don't remember. 53 to 58, I think, right? Your little grid in answers. Yeah. Maybe this one doesn't have it. I don't have the multiple choice ones on here. Yeah, this one's a little bit different. Maybe not. This is diversity index. I think I've seen this like the Simpsons diversity. This like has shown up like once. Um, I'll find one. Well, I guess these are just a bunch of uh, different calculation ones, but harder to find. These are just study questions. Yeah, I don't know, but usually, right, calculations shouldn't be too hard. They're all very basic. I say if all else fails and like you literally get what's the equation sheet here? Like if you literally get a question and you have no idea how to solve it, right? And it's a calculation question, I would just go to the equation sheet and look for the one with um the correct like units. Yeah, because like if you don't know anything, it's better than to just leave it empty, right? Because they do a kind of good job of like defining variables, right? So if you really get stuck, just look at this and kind of um, plug it in, right? They do like to ask surface area and volume questions. Hopefully you guys get one of those. And yeah, I don't know. Or posting. Any questions, you guys? Yeah. You guys are all tired. You guys have lost wait, it's lost 1 hour, right? Daylight saving. I feel like I don't yeah. No questions? Then if you have your answer posted, then you're allowed to leave. but no answer posted. You can't leave. I'll be watching you. Bye. Bye. Are you taking physics too? Huh? Oh. I don't know. Like sometimes like the university take like everything.